students, as I have promised you earlier to do EmSAT sample questions from the MOE, and that's what we will be doing today. So make sure you like and subscribe down below and turn on the notification bell to receive many more videos like this one from Mrs. Biology. Let's begin. Let's start with the first question. Which of the following macromolecules contains phosphate group? They gave us the 4 biomolecules, starting with a nucleic acid, DNA, and RNA. Let's see the structure. It's more than clear that we have, yes, the phosphate group, PO4-, but let's remind you with the structures of the rest. Carbohydrates, only carbon, hydrogen, and oxygen. Lipids, glycerol and fatty acids. But maybe some of you will ask me, what about phospholipids? From its name, we have the phosphate group. I'll tell you that's right. But the phosphate, as you mentioned, phospholipids, lipids structure something, and the phosphate only to adapt the presence of water, polarity, only in the cell membrane, I mean. Okay, moving to protein. We all know that the amino acid is the monomer for the proteins. So amino acid with amino acid, blah, 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 to connect and make the polypeptide chain. So where is the phosphate group? What does that mean? That means the right answer is the nucleic acids. The second question. Which of the following statement is correct regarding the induced FET model in enzyme-substrate interaction? Choice A. The structure of the active site of the enzyme is changed by the substrate. That makes sense. Maybe this is the right answer, but let's read the rest. B. The enzyme directly binds to the substrate to form an enzyme-substrate complex. This is 100% statement. This is right. But is it related to the induced FET model? Actually, no. C. The shape of the enzyme is changed by the chemical reaction. How come? It's a catalyst. That means reusable. So if that happened, so we cannot use it again. That's wrong. D. The active site of the enzyme changes its shape to accept a substrate. Why to change? We have for each substrate a specific enzyme. I doubt this is the right answer. So if we go to choice A, before that, let me display for you this animation of induced FET. It's the same shape of substrate, but adapting it to FET only from its name, induced FET. So if we want to choose the right answer, it's A. The structure of the active site of the enzyme is changed by the substrate. Let's read this question. Cystic fibrosis is a chronic and frequently fatal genetic disease of the body's mucous glands. It is caused by an autosomal recessive mutation. As simple as that. Please write it down. A small, B small, B small. It's very simple. I mean the genotype. In the European population, 1 out of approximately 10,000 babies is born with the disorder. That's clear. What is the frequency of the cystic fibrosis allele in the European population, assuming the Hardy-Weinberg conditions are met? These conditions means what? That the genotype frequencies in any population remain constant. And he gave me already what? The recessive, that means the Q squared. Come with me to do the work. Let's do the math. He gave us in the question the Q squared when he mentioned that it is autosomal recessive. So I chose the letter B small, B small. It's up to you again. So Q squared here equals what? He said 1 out of 10,000. So 1 divided by 10,000 equals what? 0.0001. Great. Now, he asked for one allele, which is a Q. I have a Q squared and I need a Q. So the square root of 0.001 is 0.01, as simple as that. This is the solution. So the right answer here is A, 0.01. Let's read this simple question. Each alternative version of a gene is called, I think all of you will answer me an allele. That's right, but I like to display all the time something. So the different forms of the same gene, as you see, this is an allele and this is another allele. What about a centromere? A centromere is a constricted region, okay, that join the two sister chromatids and also help in the cell division when it's attached with the fibers. Moving to a code. What is a code? It's a sequence of nucleotides. Regardless, I'm talking about DNA or RNA. Moving to chromatids. Chromatids, identical threads that when they join, they will build the chromosome. So the right answer here is an allele. Choice A. Let's read this question. Which of the following cell structures is considered as a factory for protein synthesis? Without looking at the choices, directly all of us will say ribosomes. But it seems that we don't have this structure in our choices. Let's see. A. Rough endoplasmic reticulum. That makes sense. This is the right answer because we all know that on this endoplasmic reticulum we have the ribosomes, and the ribosomes considered as the factory for protein synthesis. Good. Before choosing A, let's read the rest. Golgi apparatus. Golgi apparatus used for processing, further processing for a protein to transport it outside and other functions. Move to C, mitochondria, the powerhouse that produced energy in a form of ATP. So the last one, nucleus, the main function is to store the genetic material, I mean DNA. So the right answer here is rough endoplasmic reticulum. This is a simple question actually. The diagram below represents a cross-section through a plant root. Directly without continuing, you have please to memorize the differences between dicot and monocot, and once this is really fixed and arranged, that means this is a dicot. Okay, a student is given samples of a fluid that is rich with water. From which tissue does this fluid come? Directly, we will know it's a xylem, two vascular bundles, xylem and phloem, and phloem for nutrients and other molecules, xylem for minerals and water. But if you do not know the difference between dicot and monocot, how to differentiate where's the xylem, the phloem, you will not answer this question because they're just labeling it. So because it's dicot we know all the time it's arranged. This is first. Secondly, the xylem at the middle in the root. So A is the right answer. B, actually, it's the phloem. C, it's the root hair. And D, the epidermis. So the right answer here is A. Let's do this question. The following pedigree represents the inheritance of an autosomal recessive syndrome in a family. Here, they mention that it's autosomal recessive. What does that mean? If we want to represent the genotype, it should be in a lowercase. A small, B small, B small, R small, R. It's up to you which letter you want to choose. Now, here in the pedigree, we have these two generations. The first one, mom and dad, all of them are normal. The second one, 2 kids out of 3 kids are affected. Now, if the affected male in generation 2, they meant this, marries a normal female who is homozygous for the trait, normal and homozygous. So it's not, if I'm talking about the genotype, for example, A capital, A small, no. They mention it's homozygous and normal. What does that mean? A capital, A capital. Okay, for the trait, what is the possibility to have an affected child? Don't answer it in this way. Directly with your pencil and paper, in less than 1 minute you will find the answer. Come with me, I'll explain it in details. Let's answer the question. As they mentioned that the affected male married from a normal female. Let me represent them by letters. I mean the genotype. For the male, sure, lower cases, because this syndrome is autosomal recessive, 2 lower cases. And the female, they mentioned for us normal homozygous. What does that mean? A capital, A capital. uppercases. And any trait, they will pass it to their kids, half of it from the dad and half of it from the mom. So their kids will carry A small and A capital. Now, for me, I prefer to write it in this way, both of them the same. So here we have the heterozygous. What does that mean? The kids are not affected. So the percentage to have a child with this syndrome is 0%. So the right answer here is A, 0%. This is a good question to solve. A scientist is studying the phylogeny of 4 species, 1, 2, 3, and 4. To this end, she sequenced a fragment of DNA and she obtained the following alignment. As you see here, the sequence of DNA nucleotides for each species. With this alignment, she constructed a phylogenetic tree using the parsimony method. Which type of method is this? It's a method that minimizes the number of changes on a phylogeny. We will see how we'll do that. Which of the 4 phylogenetic trees below is supported by the DNA sequences? Okay, come with me, we will explain it in details. Let's match the sequence. Now, starting with 1. We have adenine, adenine, thymine, cytosine, guanine, guanine, and adenine in a simple way. Here we have adenine only in a 3. Here we have adenine with also with a 3. We have here thymine only with, not only actually, 3 and 4. We have thymine. Here we have cytosine only with a 3. Here we have a guanine, nothing with anyone. Here we have a guanine, we have it with a 3 and 4. Here we have adenine only with a 3. So if we want to count how many nucleotide matches with 1 and a 3, we will find 6. 1, 2, 3, 4, 5, and 6, which means that 1 and 3 are really close and related. But between 1 and 4, actually no. See the differences, only just 2 matches. Like species 2, we have here A, only 1 nucleotide. What does that mean? Let me go back to the question and I'll come back here again. As I explained there, that 1 and 3 should be very close and related. So we can exclude option B and D, and we should focus on option A and C. How we will decide which one? We have to focus with 2 and 4. Are they closed like 1 and 3? I mean sisters, taxa sisters, or not? Come back with me. Now let's match 2 and 4. Now we have here cytosine, cytosine, adenine, adenine, guanine, guanine, thymine, thymine, cytosine, cytosine. 1, 2, 3, 4, and 5. 5 nucleotides matching each other. That means they are really related. But I don't know. Can I consider them as a taxa sister or not? What does that mean? Shall I choose option A or C? Who will decide if I compared 2 and 3? Okay. If they are related, that means I have to choose A. If they are not, I have to choose C. Let's see. Let me change the color. I'll choose the red one. Here we have A with A matching. No, no, yes, T with T. And that's it. Do you think 2 out of 7? It's good enough to be close and have one common ancestor? Let's go back to the choices. I'm here again. Now I am comparing A with C. As we mentioned, I'm really confused if I want to consider 2 and 4 as a taxa sisters or not? Who decided the relation between 2 and 3? And if you remembered, okay, I found two nucleotides matching each other. So they are related. So we can consider them that they have one common ancestor here. They are not too much related. That's why we are supporting the answer to be A. So the right answer is A. Now, a student immersed a small piece of potato tissue in a 0.25 mole per decimeter cube sucrose solution, and the tissue showed no change in mass. Good. What would the student find out when he immerses the piece in a 0.1 mole per decimeter cube sucrose solution? If we just compare the concentrations, we will find out that he added water here. That means we increase the water potential here. So what will happen? Let me display this figure that will simplify the whole concept. Now, on the right one, on this beaker, you will find that we have a rich sugar solution, high concentration of solute. That means low water potential outside the potato. So, from the potato slice or the tubes, the water will leave and go to the solution outside because the water potential outside is less than the water potential inside the potato tube. Here, the opposite. If we put it in pure water, that means the water potential outside is higher. I mean the concentration of water. So it will start move from outside to inside. What will happen with the potato cylinders? Will gain more mass. So we answered the question. First option, the mass would have increased because the water potential of the cells has decreased. 100% right. This is right. But let's read the rest. B, the mass would have decreased because the water potential of the cells has increased. Sure, this is wrong. C, the mass would have increased because the water potential of the solution has decreased. We mentioned that we added water here. We decreased the concentration, so also this is wrong. D, the mass would have decreased because the water potential of the solution has increased. Again, this is wrong. The first statement is wrong. So the right answer, as I mentioned, is A. Now this question. The ecological relationship between two different species is shown in the graph below. And this graph showing us the population size against the time for A and B. What ecological relationship does the graph represent? Directly, I want to exclude B and D. Why? B, extinction. If that's right, at least you will find one of populations disappear because it's extinction or both of them. So B is wrong. D, parasitism. One will get benefit and the other will get harmed. So one will increase and the other one should decrease. So that is not there. So D is wrong. Now A and C, mutualism and commensalism. I will tell you I'm supporting A. Why mutualism? Both of them will deal with each other, will get benefits, so both of them are increasing at the same rate as we see. But what about commensalism? Maybe you'll ask me, commensalism one will get benefit and the other one will not be harmed. That's right. But why it's not commensalism? In commensalism, once the one, for example, B will get benefit from A, the rate of increasing should be higher than A. But as we see, the rate of increasing for both are the same. So A is the right answer, mutualism. Let's read this question. The diagram below represents a food web in an ecosystem. If the population of hogs here in this area increases, what effect will this increase have on the web? Let's read the options. The first one, the prey populations of hogs might decrease later, which is right 100%. Okay, the other populations in the web might decrease. Sure, when we have a limited resources now, it will affect the whole web. But let's see the other options. The prey populations of hogs might decrease. Again, we agreed on this. Later, the hog population might remain unchanged. Actually, no, because once we have a limited resources, the number of hogs should decrease. It will be affected. So this is wrong. 3. The prey populations of hogs might decrease. We agreed on that also. Later, the hog population might increase. Again, this is wrong. It will decrease. Now, the 4th option, the prey populations of hogs might decrease. The same right statement, but later the hogs would find another prey population. Logically, that's right. But in this food web, it's not mentioned. So I cannot consider the statement here is right. So the right option should be the first one. This is a question. The chart below shows the messenger RNA codons for different amino acids. Given the following codon sequence, as we see, we have 7 codons from 5' to 3'. Which of the following can be described as base substitution causing a nonsense, please focus here, mutation in the messenger RNA sequence above? The whole story, what is a nonsense mutation? In a simple definition, it's a premature termination for a protein. What does that mean? After we receive the messenger RNA, after transcription, we need to make the translation. So normally, if we translated the 7 codons here that appears, we will receive the right protein. But out of sudden, substitution happens somewhere and stop the translation, not after finishing the whole 7 codons. This is the nonsense mutation. Maybe stopped here, substitution happened. Instead of bringing the normal amino acid, we brought the stop codon. Let's see which option is the right one. I'll move randomly with the options. So they are talking about substitution happens with UAC, here the 3rd codon. Instead of UAC here in option D, we substituted by UGC. Directly, I will go to U, G, and C. No, this is cysteine, so this is normal amino acid, not the stop codon. So it's not D. Moving here, UCC. U, C, C. It is serine. Again, another normal amino acid. That does not mean it's not a mutation, but it's not the nonsense that I'm looking for. Let's see here, UAU. This is U, and A, another U. Tyrosine, another normal amino acid. Moving to option A, to UAA. This is U, and this is A. Where is A? I found it. This is the stop codon then. So the right answer is A. This is the last question. Which of the following could not be considered a density-dependent factor affecting population? It's about ecology. Let's see. Climate temperature. What's the relation between this factor and the size of population? Too much or very little? Climate is a climate. But let's move to the rest. Limited in nutrients? Sure, that will affect the population size. Build up of toxins? Again, maybe from water, maybe from food, and again, that depends on the population size. Predation, the same. Just if we look at this, we will find some factors between dependent and independent, and we will see here predators, diseases, competitions, and for independent, the habitat, hunting, something not related to these populations, the natural disaster, and we can add to that the climate. So the right answer here is A, the climate temperature. Now we have finished these questions, but don't worry, I still have more sample questions coming up for you students to watch. So stay tuned to receive the next part of these EmSAT sample questions by liking, subscribing, and turning on the notification bell down below to receive the next upcoming videos from Mrs. Biology. Thanks for watching.